talked about our catabolic metabolism. Now I'd like to talk this week about protein synthesis and DNA. And we are jumping around in the book a little bit, but we're trying to follow the same schedule as the other sections of the class. So let's think about DNA. First off, when we see this shape, this X-shaped thing, the reason that they show it to us in that shape is an artifact of how we can visualize the chromosomes. So DNA is very long and very thin, and if we're in the middle of the cell cycle and we're looking at under the microscope, we can't see the DNA. We can see the nucleus, but we don't see the DNA. But just prior to cell division, the chromosomes have replicated and become dense. And at that point, we can visualize them, and we can see them in a shape like this, in this X shape. The chromosomes are only in that shape right before the cell is going to divide, and they will separate right at this point. So this representation of chromosomes is a little bit misleading, and the reason that it's there is that's how we can see them under the light microscope, right? So just bear with us and see that they're trying to represent the DNA. And so what we want to notice here are these nucleotides A, T, G, and C. A is always going to bind with T. T is always going to bind with A. G is always going to bind with C, and C with G. That's how it works in DNA. In RNA, we don't have the T, we've got the U. All right, so that's adenine, thymine, guanine, cysteine. All right, and so here we're looking at what we call a sequence. So this is the sequence. Notice that it's a template. If we know this strand is going to be complementary. So if we have ATG up here, we have to have TAC here, right? Because these are going to match up. These two strands are going to bind with one another. Those are hydrogen bonds, right? These are hydrogen bonds between those nucleotides. A and T have 2 hydrogen bonds. G and C have 3 hydrogen bonds between them. So that allows us to open up the DNA and read sections of it. So in this portion of this illustration, we're just looking at the sequence. And here we're seeing this helix that is formed by the chemistry of the molecule. And of course, it's very long. The molecule is quite long. And then here we're seeing the DNA wrapped around some histone proteins. So these histone proteins act like a spool, right? There's something that we're winding around. We're using it to wind the DNA around it and keep the DNA from getting tangled. All right, so we've wound it around these, and we call that a nucleosome, a nucleosome. Once we've got the DNA wrapped around that histone protein, we call that little bead-looking thing a nucleosome. And then we kind of condense that, kind of put it into a little coil, and then put that coil also into a coil. And so all of this is condensed into a chromosome in a way to keep it from getting all tangled up, particularly during cell division. Right, so this is what it looks like during cell division. In this illustration, they've attached it here, but don't worry about that attachment. For when we're talking about protein synthesis, we're pretty much interested in this sequence. And another way of thinking of protein synthesis, some other words for it could be gene expression, also transcription and translation. So those are the 2 stages. Transcription and translation are the 2 stages of protein synthesis. And gene expression is what protein synthesis does for us. We're reading a gene and expressing it. So pretty much one understanding of our genes is that they code for a protein, and then that protein does something in the body. It either acts as an enzyme or a hormone or something structural or something in defense. All right. So here's another look at those complementary strands. Here we're seeing C and G with their 3 hydrogen bonds. Here we're seeing our A and T with the 2 hydrogen bonds in between, and these different colors are representing the ladder inside the DNA helix. The things to note here are that we've got a complementary strand. So if we've got one strand, if we only can see one strand, let's say we see A, C, G, G, T, A, then we can easily tell what is that other strand got to be. What is our complementary strand going to be? It has to be T, G, C, C, A, T. That's what we mean by complementary strands. So if you know one strand, you can determine the other. Same as the cell does, same as the enzymes in the cell. All right. So this is a representation of DNA replication. So DNA is replicated, and again, this is just totally goofy, right? This happens after, right? That's what we see after this replication. So when we're replicating DNA prior to cell division, we have an enzyme here called a helicase, which opens up those two strands, right? Breaks that hydrogen bond, makes those two complementary strands available out there in the cell, and the cell makes a complementary strand. So here we see DNA polymerase, which is making that second strand. So it's making that second strand to fit, to be complementary with that first strand. Okay. Now the thing about DNA polymerase is it only works in one direction, right? So on this side it's doing great, right? It's just zipping along. Well, on this side the DNA polymerase is going the other direction, but the strand is only opening up a little bit at a time. So it works for a section, and then it has to leapfrog back and make another section. And so these are little fragments, little DNA fragments that get added, and we have to have some other enzymes come in and link all of this together. I'm not too worried about that at this point. I do want you to know that when we are replicating DNA, and we've got these two new chromosomes that are still stuck together, we call this semi-conservative. We're saving this strand, right? This original strand, we just put a new strand next to it. And this old strand, we put a new strand next to it. So this is called semi-conservative, right? Each daughter strand is going along with a parent strand. All right, now let's think about the genetic code and a cell's protein. So some things to know here are that all organisms use this same code. What do I mean by a code? We mean that this set of 3 nucleotides codes for an amino acid. So this is methionine. It's our start codon. It's our first amino acid. So where did we get all this? Here's our template strand. This is our DNA, our double-stranded DNA. We've got TAC, we've got ATG, our RNA, we've substituted that T for a U, and that codes for methionine. Well, we've left a whole lot of steps out, right? But we're showing the flow of information, but we've left a whole lot of stuff out. So actually in a given DNA molecule, either side, depending on the gene and what's being expressed, either side might be the one that's going to code for the protein. So when we're coding for that protein, we're going to open this up. We're going to make a template. We're going to copy off of the template strand, and we're going to get a messenger RNA, an mRNA, messenger RNA. Right? So notice that this messenger RNA is copied off of this template strand. So there was a T right there, so we've got an A. There was an A right there, we've got a U. So remember, in RNA, instead of using thymine, we're using uracil, so we've got U. U is just where we would find a T. All right, so there's a C up here, so there has to be a G here, complementary. All right, we've got G, so there has to be a C. G, so there has to be a C in the complementary. C, We've got G on the complementary. All right. So you're good on the complementary, right? And so this is our messenger RNA. This is produced in the nucleus. It's carrying the information from this DNA. It's going out of the nucleus, and the ribosomes are going to grab onto it. And wherever it finds AUG, the ribosomes are going to stick the amino acid methionine in the polypeptide chain. So next, we've got CCG. That's the codon for proline. Next, we've got CAA, and here's our amino acid. So this is happening. The ribosome is doing this. Ribosome. Okay. And it's creating this as a chain, right? This is rolling off as a chain. And there's probably some good graphics on this on YouTube. I'll try to post those for you. All right. So that was kind of the overview. So here we're having a look now at an opened up DNA. And again, this is not like that. It's not like that. Right? Cover that up. No. All right. So we're opening up that DNA where we want to express that gene. We have an enzyme here called RNA polymerase, and that's just making an RNA polymer. And so there you see your RNA transcript, just like we talked about. It's complementary to this DNA strand. So this is our messenger RNA, right? That's our messenger RNA, or mRNA. And this is happening inside the nucleus. This messenger RNA is going to leave the nucleus and be translated. This is transcription. Transcription is when we make that messenger RNA using the DNA template. We're making it out of nucleotides. We're using U wherever there would have been a T in DNA. And we're calling this our messenger RNA. The whole process here is called transcription. It's happening inside the nucleus. All right. We have some other things that we can do inside the nucleus. We can cut out some bits, and we can add some things on either end. In this case, we're cutting out the introns, and we're going to keep the exons. Exons are expressed. This intron will just go out and get, recycled or something. All right. So we've spliced that long chain of information and spliced it up. And there's a lot here that we're not going to get into in this course, but this is part of gene expression. The same strand or sequence of DNA could code for different things based on how we do this post-transcription modification, right? What gets cut in and cut out. That's genetics, so don't worry too much about that. But exons are expressed. That's a mnemonic for that. Exons are expressed. These are spliced together. This is going to leave the nucleus and be turned into a protein. All right, translation from RNA to protein. Translation from RNA to protein. So during translation, that messenger RNA is read by the ribosome, and we're also going to use these tRNA molecules to do that. The tRNA brings the appropriate amino acid. T stands for transfer. So the tRNAs are transferring amino acids. So here we have an anticodon. Here's our tRNA. There's our amino acid. So let's look here. This is our messenger RNA. These are our ribosome subunits, and they're just grabbing right onto that messenger RNA. There's our start codon. AUG is always going to be our start codon. It's going to code for methionine. So if our start codon is AUG, the anticodon, or the thing complementary to that anticodon, has to be UAC, right? UAC is going to be complementary to AUG. So this anticodon on the transfer RNA actually comes up and gloms on there, and there's our methionine that's to start. All right, so now we've got our first transfer RNA and protein there on our ribosome. And our next codon is UUU. So what has to be our anticodon? Has to be AAA. So here comes our next transfer RNA, AAA, and here's our amino acid phenylalanine with that. So now we've got two amino acids. These, we're going to have a bond right here. We're going to have a peptide bond, right? Peptide bond between these two amino acids. And our first transfer RNA is going to drop off. The methionine will remain attached, right? So now we've got our first two amino acid. Here comes our next transfer RNA. And same thing, we've got a codon, anticodon, and amino acid. This will slip in. We'll have a peptide bond between our two amino acids, and these will move right through. This actually happens pretty quickly. Right? So remember, this is our transfer RNA, messenger RNA. These are amino acids. Transfer RNAs are transferring amino acids, and they have the anticodon on them. Right? All right. So here's another overview of that. So we've got our DNA, and through the process of transcription, we're going to make a messenger RNA. That messenger RNA is going to leave the nucleus, and the ribosomes will grab onto it and use that information to make a protein sequence, which is just a straight chain of amino acids, right? And then there can be further modification of that protein, can make into some different shapes. All right, there will also be some videos from online resources to go over this material. And feel free to let me know if you want to meet and have a Zoom on this material.